Thank you for the introduction. I am Eunyong Kim I'm a member of the team KBJ. I'm gonna present the work we did for the preterm birth prediction challenge. So this is the table of contents for today's presentations. Before moving on to the work we did, I'm gonna briefly introduce the preterm birth. The preterm birth is important because around 11% of the pregnant women actually gives the preterm birth. So, and also it can lead to some various diseases, including the breathing problems or the blood problems, and even it can lead to the death. So it has been a lot of researches on the factors of the preterm birth. Of course, there are a lot of factors. Among them, the increase of the diversity in vaginal microbiome can lead to the preterm birth. So there have been a lot of researches, and some microbiomes, some microbiota were known to have some correlations with this preterm birth. So, as many presenters already showed that there are mainly two tasks in this preterm birth prediction challenge. Since we won the second place for the task 2, I'm gonna focus on the results of the task 2 prediction of the early preterm birth. But we followed the same pipeline for both tasks. This is the overview of our approaches. There are mainly three steps: the data pre-processing, feature selections, and the model constructions. I'm going to go on the details in the next slides. This is the summary of the data sets we've got for this challenge. There are around 3,600 samples and 1,300 individuals. And we did some data pre-processing, filtering by the collecting weight conditions, because to make this data to be under the same condition as the test data set. And because the model was validated at the level of the participants, we averaged over all those samples to the participant level. And you can see the used features under here. We used all those data retrieved from the Mulliamp pipeline except the pairwise distance. And we used, we used the race data from the metadata set. You can see here, there are two challenges that make it hard to predict the early preterm. The first one is that the data is very imbalanced. And the second one is that the size of the dimensions of the features are very large compared to the dataset size, because the total number of the features that collected from these feature lists were more than the 50,000. So to overcome this problem, we did the feature selections. There are many methods for the feature selections. Among them, we applied this minimum redundancy, maximum relevance, MR approach. It is quite simple. Just pick the first feature, and then iteratively select the other features to make these features to be less, to have less redundancy, but to maximize the relevance between the feature sets and the Y labels. So we did this until the total number of selected features to be 300. And we applied this feature selection method to 3 types of features because the dimensions of these 3s were too high. And the second approach to overcome the limited data size, we constructed an ensemble model. Firstly, we used the lazy predict Python package to construct 26 different machine learning based models. And of course, we could rank the model by the model performances through the fivefold cross-validations. And then we selected the best performing models that ranked in more than two-folds. After that, we constructed ensemble models using the selected models. So I'm going to show you the results here. The first one is that the impact of the feature selections. We draw these PCA plots, and you can see that after the feature selections, the data sets could be discriminated. You can see the samples are very close together before the feature selections, but you can see that you can see these selected features can discriminate these early preterm and term samples. Also, we constructed the model, and you can see the results from the fivefold cross-validations. And of course, without the feature selections, the model showed very random performance. The AUC values were around 0.5. And this is the actual results of our model. Because we could not get the test data sets, this is the result of the cross-validations. And this blue line is the performance of the ensemble model. And the rest are the single machine learning model that used to construct the ensemble model. You can see that by combining all the other models, it performed very well. And this is the result of the test set. You can see the values on the website. And the point of the test set performance is that our model showed very balanced sensitivity and specificity, although the data sets were very imbalanced, and we did not use any approaches for the undersampling or oversampling the data. We can say that the model can predict the both positives and the negatives data as well. And the last thing is the feature interpretation. It actually showed very interesting results. We looked into the top 10 features, top 10 the taxonomy features. Actually, we used the taxonomy features at the genus level. And among the top 10 features, we could actually find the known microorganisms in preterm birth through the literature search. And these green are the known organisms, and the blue one is the genus that were related to the vaginal microbiota, but not the preterm birth. And we expect these red organisms, or maybe the new microbiota that can be related to this preterm birth. And likewise, we looked into this top 5 CST features, and it showed the similar results through the interpretation. We found that this lactobacillus and the genus Prevotella are highly related to the preterm birth. So in summary, we used these two approaches, feature selections and the construction of the ensemble models to get the high performance. And the model showed our model actually ranked in the second place in task 2. And since because of the limited number of the submission, we could not test all the hyperparameters and hyperparameters. So the model can be improved with the more elaborate feature selections. So this is the end of my presentation. Thank you for the Dream Challenge organizers, and thank you for my team members. Our team members are from 2 different institutes, Korean Bioinformatics Center and the Bioinformatics and Intelligence Lab in GIST. Thank you.